Once we are live, we will get started. Okay, so welcome back to day 2, part 4, our last session for today for the workshop. Last session of the workshop for this session. So thanks for sticking with me. We are going to be talking about phylogenetics at this point, and so ideally, while it says phylogenetics of metagenomes, really what we're hoping to do is work with these individual genomes that we've pulled out. So the ones that we've worked with now for through the assembly method, through the binning method, through some type of annotation, we're now going to be doing some phylogenetics, and we're going to try and figure out where these organisms are on the tree of life. So the outline for this part of the talk is going to be a short introduction about phylogenetics, and then we're going to get into how to build a phylogenetic tree using concatenated ribosomal marker proteins. So we're going to be continuing on from the last section where we use HMMs to find the models for ribosomal marker proteins, and then we're going to go a step further and actually do the construction that we need to do to make a tree that uses all that information. And then I guess there'll be a discussion at the end of it because it says so on the outline. So why is this important? First and foremost, we want to, when we have genomes that we're looking at, and maybe we're having a hard time understanding where they are taxonomically based off of similarities in databases, we want to be able to decide where they are on the tree of life. And so we can take this and we can take novel taxonomic groups, novel lineages, or metagenomic bins, or anything that we're looking at, and we could put them in greater context on a phylogenetic tree of all the organisms that are available out there. And so understanding evolutionary history is really important. It gives us ideas about speciation, and when we compare that with things like geography and the age of the taxa, we start to see these patterns that emerge that we all are kind of very familiar with. The idea of endosymbiosis for the algae that are out there, how the tree of life has developed. There's this active debate now that really getting a little heated, depending on who you ask, about the accurate placement of the archaea versus the eukaryotes and how they are related to each other, and if one is an offshoot of the other. And then the last reason that we can use phylogenetic trees and what we can do with them is that we can actually use it as part of the process that we were talking about just previously with annotation of more accurately predicting function of novel genes. And so the example here I have is the DSR gene, where based off of its position in a phylogenetic tree, it actually functions in a forward or reverse process. And so knowing which part of the tree it's on, despite what annotation you might have, will be important of telling if it's if the organism you're working with can oxidize or reduce sulfur compounds. So here's an example of the 3-domain tree of life from Carl Woese in 1990. So we can see here that life on Earth is very simple. There's only a couple major clades of bacteria and a couple major clades of archaea, and there's some eukaryotes up there too. I guess they do things that are pretty interesting. So we're probably done 1990. We're all fine. We don't have to worry about it anymore. Oh, wait. 2016 came along, and we have this nice tree generated by Laura Hug et al. In the Nature Microbiome that year, which showed what a full tree of life may look like. More accurately in terms of where the bacteria are and where they are in relation to the archaea and the eukaryotes. And not only on top of that, but they illustrated that there are a lot of lineages that do not have any cultured representatives. So every red dot you see on that tree is a branch of the tree of life that we don't have any cultured representatives to work with. And so to give you an idea of what we need to do to get our starting data into a nice tree that looks something like that. What we need to do is we need to identify our marker proteins or marker genes in our genomes. We need to perform some type of an alignment. We need to perform some type of trimming step to make sure that we're working with the best data that we have. We then take those alignments and can concatenate them together. We can then construct a phylogenetic tree from there, and then we have to visualize the results because we can make a tree, but you got to look at it, right? And so there are lots of ways that we can look for marker genes. here. And so we can look for them in DNA, RNA, or protein space and make comparisons based off of that. And so in non-coding DNA regions, so this might be some of interest of people out there. So areas that aren't in protein coding regions tend to have a higher mutation rate. So if you have two closely related organisms, you can look at these regions and map out a phylogenetic tree of these regions to see how fast certain elements of those, some elements of closely related organisms, how rapidly they're evolving relative to each other. In proteins, there's a much lower overall mutation rate. And the reason for this is because, and ideally, if you're an organism that likes living, you want to maintain the functions of the proteins that you have in order to continue to thrive in the environment that you're part of. So proteins can be really good for distantly related organisms. And there's a reason why we use proteins on the phylogenetic tree, the big one that Laura generated. Dr. Hug. Okay, so there are lots of potential markers that we can work with. We've been talking about these almost ad nauseum for the last couple of days. 16S rRNA, which is really good because there are hundreds of thousands of environmental sequences, so we can map our rRNA genes onto organisms that haven't been identified yet. There are lots of well-developed trees, things like Silva, which generate a tree every time they release a new version of it that can be visualized in ARB, which is an older program and can be a little finicky to work with, but can be a way to look at a lot of 16S data all at one time. Bad news is that 16S tends to break assemblers when we're working in metagenomic data sets. And so if you go through and look at genomes that are being generated from metagenomic data sets, they have a tendency not to have the 16S gene as part of them. And on average, I think, with the data sets that we work with, it tends to be about 40% of the genomes have a 16S and the rest of them do not. And that's because, as we discussed in the assembly part, 16S have lots of areas that are conserved, and those conserved regions lead to bubbles, and bubbles lead to things that can't be resolved, and then the assembler breaks on them.
And many genomes have multiple copies of the 16S gene.
And that also makes things complicated when organisms have multiple copies of the 16S for the same reason. It also is generating more bubbles there as well, not just on the conserved. So we have two elements that are happening there just to highlight this. You have bubbles occurring because organisms have conserved regions. And so similar organisms or organisms that are related to each other might have conserved regions that create bubbles and they pop. But also within a genome, if there are multiple copies of that 16S, it'll create a bubble where the assembler cannot resolve which direction certain fragments of the genome are going, and it will also pop there. So we can also use ribosomal marker proteins or concatenated ribosomal marker proteins. And we concatenate them because each individual protein gives us a story, but there is a limited amount of information there. So we can concatenate them and go from something that's about maybe 100 amino acids to a couple thousand amino acids. And then that way we have a lot more information. There are many more sites that we can ask information on a phylogenetic basis on that. But then we run into this problem of, well, this is only going to work for things that we have genomes for, right? So we can't go into the environment and get all of the related ribosomal single copy marker genes from organisms without having them in a genome first, right? So if your goal is to find, are there environments where your organism, which is unknown, has been seen before, 16S is really useful. But if you're trying to figure out where does this organism belong on the tree of life that we've seen in terms of genomic diversity, ribosomal markers become really useful there at that point. And so we've talked about this a couple times in passing, so let's talk about it in detail. So there are different types of ways that we can perform alignments. So there are local alignments where we're trying to optimize small areas of a sequence comparison, and we're basing scores off of these much more limited areas. And that's really what matters when we're getting a score back from it. And then the other option is a global alignment, where we're trying to find all areas between two sequences that might match up, including regions where we might have to add gaps or remove gaps to figure out where the information is the most accurate. And so these global alignments allow us to take things that are a little bit more divergent from each other and find out where they are. And the more sequences you can fill in there, the easier it is to make those types of global alignments. And a third type of alignment, which we're not really going to get into really at all too much today, are these structure guided alignments. And so we use the really gold standard example of this is the ARB data set that comes with Silva, which allows you to take a new 16S sequence, align it to their data, and then look at it visually, and they have that visually broken up so that you can actually see where the 2D structure is of your rRNA gene. And so if you're looking at the alignment and it looks like it's a little off, and they have all these symbols that tell you if it's, it correctly fits into a loop or it correctly fits into a stem, you can make little adjustments based off of the structure of the rRNA to get a more accurate alignment. So the easiest thing and most problematic thing about an alignment is you can make an alignment of anything as long as you're willing to make add enough gaps. And so we have to put some limitations based off of the scoring algorithms that use to make sure that we're not just adding gaps all willy-nilly in a way that doesn't have any actual biological meaning. And so this is just an example of a short amino acid stretch comparing hypothetical chimp, human, gorilla, and orangutan sequence. And you can see with these columns here that there's actually a bunch of different ways of sorting this information with gaps and missing information to try and make an alignment. And so in this first version of it, we use the R as we use the R as the marker between the humans and the chimps. And so we try and align everything that way, but that leaves a gap in the gorilla and the chimp sequences. We can move it over because the gorilla has an S in that position and the chimps don't, so we can move that over. Now we have a gap, or we can have a gap situation that looks like this where we can move it around. And you can imagine this gets pretty elaborate once you start having, 4 or 5 or 6 or 12 or 20 or 25 amino acids that have been added into the sequence, and we need to figure out where they belong in relative to other sequences. And so it's possible to align 2 different sequences, a bunch of different, and get a lot of different gap results. Here's another example. So aligners want to be your best friend. So if you give them information, they're going to do their very best to do the work for you. And so you can get an output that looks something like this, where you have lots of alignments, and then it gets to this point where it goes, I don't know what to do here. And it has this little short fragment and just sticks it there. This is the most parsimonious result this aligner could come up with, but it really doesn't have any meaning, right? There's no extra information there. It's missing a lot of the data. And even though we found the sequence, maybe it was in a database, maybe we were sure it was 100% what we were looking for. If you see something like this, it's not actually useful, and you can't use it to build a phylogenetic tree farm. So we'd remove something like that if we saw that going forward. We can talk a little bit about the types of phylogenetic trees that we can make. So we're going to highlight a couple of them here, including distance, also known as neighbor joining, maximum parsimony, maximum likelihood, and Bayesian trees. I'm going to give you about as much information as I know about phylogenetic trees going forward right now. If you have questions, we can talk about it, and maybe I'll have an answer. But phylogeny scares me, especially in when you start getting into things like maximum parsimony and Bayesian analyses. There's too much math. I get really terrified. So, come at me very slowly if you have questions about those methodologies. So a distance tree or neighborhood tree calculates a pairwise distance between alignments. And what we're doing here is basically just counting differences. So we get a couple sequences together, we make an alignment, we figure out how many differences there are, and then we use an algorithm, a computer program to just make a distance matrix based off of that. And we can tell how far apart these particular sequences by how many changes there are in their pairwise comparison. And then we get a tree that represents that. And so this is really great because it's fast. It's a great first tree to make because it is fast, and you can get a lot of results very quickly from this. And it's really good when there's a low sequence distance, right? So in this instance, where if you have sequences that are related to each other and there's only a couple gaps, differences, a couple base pairs, a couple amino acids, you can build a tree very quickly, almost and very accurately, that would tell you what the relationship between those are. Unfortunately, when it gets to more complicated data, this is only based on one parameter, the parameter of how many changes there are between any one sequence. And so it's not really good and doesn't take into account multiple mutations or mutation sites, or taken into any consideration any of the models that we have about how often certain changes occur in DNA or amino acids.
good for closely related organisms?
Closely related sequences.
Sorry.
Yeah.
Right. Closely related sequences. Why is this fine?
So, I mean, good is a relative situation here, right? So it's good because when they're closely related, right, you already know that there's some relationship between them, right? So all you're trying to do is map out how they're different and predict in what way the differences arose, which is what the phylogenetic tree is really showing you. It's really a way to do that really quickly. I'm not saying that that's the tree that you should make and then publish, but it is a tree that gives you some of that idea of that relationship of what you're looking for. And so this is an example, a crude example of a distance-based phylogenetic tree where we have a distance matrix here that asks how many differences are there between sequences A through D. And so A has 0 differences with itself, but has 6 differences with B. B has 7 differences with C, but or B has 3 differences with C, and A has 7 differences with C. And you can take this information and you can give it to a computer program, and it can find a path that would explain all of these distance values until you ended up with a tree that looks something like this, where you take the distances additive along the horizontal to figure out how far apart a sequence is. So in this particular instance, if we wanted to ask the question of how far apart A is from D, truly, we could take the distance here, which is 5, plus 1, which is 6, plus 2, which is 8, and then we come down here, and then add this whole distance, and that's 6 plus 8 is 14, which is how many differences there are between these sequences. So we're adding the horizontal distance between 2 sequences to get an idea. So in this particular situation, it becomes one of those things, and we'll see this more and more often, is that vertical distance in a phylogenetic tree doesn't matter, only the horizontal distance does. And so that's a big thing to remember. So you can have really, really shallow branches that are very, very long on the tree and be like, oh, well, this sequence up here must be really different from this sequence down here. But if the distance that they're apart from each other horizontally is really short, that's not actually the case. It's, it's the way that we think about these trees, but it's not actually what the data means. Okay, so we can talk about a little bit about maximum parsimony. So in this situation, we use all possible trees from each position of the alignment, and trees get scored based on the number of steps needed to generate a specific tree. And so in this instance, we're looking for a tree that can be explained in the minimum number of steps. So that's what the parsimony part of this comes in, right? So we're looking for a solution that explains this in the simplest terms. And so it's nice because unlike a neighbor joining tree, where we only use the distance between 2 sequences, in this case, we're using all the data, all the positions, all the information that we have. And it's good for closely evolving, close evolutionary distances. Some of the drawbacks of it are it's slow. And a lot of these drawbacks and a lot of these issues come in based off of your computer and based off the data set size that you're working with, right? So this is always very relative. So doing maximum parsimony back in the day when computers were slow, it would be problematic to try and do this with maybe 100 sequences. Nowadays, it's like, okay, 100 sequences, we can do it pretty quickly. Everybody has supercomputers in their pockets. But if we want to do maximum parsimony for very large trees, it can take some time. And we get into that process where it becomes a little bit more inhibitive. One of the downsides to it is also that it assumes equal rates of mutations so that every site has an equal chance of being mutated as any other sites. We actually know that that's not the case. And other models and other trees making programs take that in consideration, and we'll talk about those as we go forward. Like in maximum likelihood. So maximum likelihood actually has information built into it that you get to select the model of evolution that you want to use in your tree construction. And so this can do things like assume that there's a different rate in transitions and conversions in your DNA sequences, right? So the change from an A to a T has a different rate than a change from an A to a G or a C. And we can generate models that can be incorporated into these phylogenetic tree programs that allow us to more accurately calculate evolution on a scale that is more realistic. And so each each is calculated on each position, and it represents all of the sequencing data. So that's a big positive. And it's most likely to have, this is the tree that when you're working with is most likely to have produced something that has meaningful information based off of the observed data that you might have from other locations. So it uses all the data, it incorporates models of evolution, it corrects for the multiple mutation situation, right? So if you have a position where there's a mutation, you can actually work in the possibility in these models of what's happened. Maybe this isn't the first mutation at this position, maybe it's the second or the third, and we can model that information on all positions simultaneously. And so it makes it really good for large evolutionary distances. Again, this can be a little slow. We're going to actually work with a maximum likelihood tree program that's an approximate maximum likelihood tree program, which is actually pretty fast. But one of the more commonly used maximum likelihood programs, something like RAXML, that when you get to very large numbers or very large sequence lengths, can take a long time to run, days, weeks, sometimes months. They have the number in the Hug paper, if you go to it, about how many computational days it took to generate that tree over something like, it might have been like 20,000 positions, 10,000 positions of amino acid concatenation, and then over their full tree, which is a couple thousand, tens of thousand organisms. And it was something like, you know, computational years that took, right? They ran it on a supercomputer. It took some time, even at that point. And so it can be really, you know, demanding in terms of computational prediction time. And so it's one of those things. So if the data is getting very large, you're not running it on your laptop, and you might not even be able to run it on your server, or on your server, yeah. And then last but not least, Bayesian phylogenetic trees. And so this is a model or a method for making trees that incorporates Bayesian statistics into calculations at each branch point, right? And so we'll talk about a little bit about how maximum likelihood gives you some certainty at various branch points. But in a Bayesian phylogenetic tree, what we're using here is our understanding of our any given site being a mutation site based off of the probability of other sites in the gene. And so we're using its posterior probability, or the possibility of, or the information that we know previously about the tree to generate this. And so the idea here is that we're trying to choose a tree that has the highest posterior probability, the highest probability of actually existing in the real world. The good news is it's relatively fast. It allows for very complex models and complex tree estimates and high degrees of measures of uncertainty. So that way you can get a fairly accurate tree about what you're looking for. The downside is that because you use posterior probabilities to make these calculations, you need to have some prior information going into it. And so you need to have at least a base tree that you have fairly high confidence in to start with before you can go in and make some Bayesian calculations about what you're looking at. So that's the broad overview of Bayesian trees. If you want more information, I can direct you towards it. Like I said, they scare me a little bit. But if anybody ever wants to talk about them, I'm happy to share a little bit more of what I know. So there's a lot of different ways that we can look at different evolutionary models and how they're incorporated into making trees. And so they depend on the types of assumptions that we want to make. We incorporate nucleotide and amino acid substitution models. We can incorporate codon-based models, and then models, you know, so go even far as models that depend on the secondary structure. And so when you're asking, well, which model should I use? Why do I want to use a certain model for your data? There's actually a couple places you can supply, you can get estimates for that. So these are two web pages, JModel Test 2 and IQ Tree, where you can input your sequence data, and then they will, based off of some of the criteria buttons that you can select there, will give you an idea of which maximum likelihood model would be best for your tree to run on. Great news is it's free. Bad news is that these aren't ideal for your Bayesian approaches, that they only really work for your maximum likelihood trees. So with Bayesian trees, we get information at each branch point based off of the posterior probability. For maximum likelihood trees, despite the text being a little skewed here, what we use is a tool called bootstrapping. And so with that, what we do is we pseudo sample a tree that we have constructed, and we pull out elements of it, and we ask what happens if we are missing an element. How does the tree look at that point? And so we do this a bunch of times, resampling it in a bunch of different ways. Bootstraps can range in how many subsamples you're going to make. So generally you see somewhere between 100 or 1,000 in terms of bootstrap calculation values that you're going to make. And what you get to do is you are asking, how often does a certain tree branching pairing occur? So in this example here, we've run 100 subsamples of this tree of, despite the text being like this, are of chimps, humans, gibbons, orangutans, and gorillas. And what we can see is that this branch here between the chimps and gorillas occurs 41 out of 100 times. And compare that to this branch here, where the chimps are on their own branch, and it's now the gorillas and the humans that are more closely related, happen on 28 of the 100 trees once we start subsampling them. And in this case, it's now switched where the chimps are more closely related to the humans in 31 of those times. In this instance, every time we look at a tree or re-subsample it, we actually have the same position for both the gibbon and the orangutan. They're always on the same branch points with each other for this whole tree every time we look at it. So when we get to this point of saying, well, what does an actual tree look like? If we want to look at one tree, not 100 of them, how do we do this? What we do is we take the tree that has the highest bootstrap values for all of the points, and then we overlay those bootstraps values so we can tell how confident we are of any one position. So in this instance, in this final tree, where the letters are nice and easy to see, we can see that because the gibbon and orangutan were always next to each other, they have a bootstrap value of 100. So they occurred in 100 of the 100 trees. When we ask the question of where do humans and chimps and gorillas relate to each other, the one that had occurred the most times in their subsampling was this chimp versus gorilla split. It occurred 41 times. So that's the one that we're going to use on our final tree. But 41 is not really that great of a number. I don't know if you noticed, out of 100, it's only 41%. It's not great. So a lot of times what you see are people displaying trees in your manuscripts or in your poster figures or something like that, presentations. They generally only highlight areas that have bootstrap values, sometimes higher than 50, but sometimes higher than 75, and sometimes even higher than 90. And so those, the higher you get on that score, the more confident you are about a certain split on the tree, and the relationship that occurs between those organisms or those sequences or whatever you're looking at. And so generally, if you are making claims about phylogeny or how things are related to each other, and you have a branch point that has a really low bootstrap value, and that's what you're basing all of your research on, you are probably going to get pushback from reviewers as they look at that and go, well, that's not really that strong of a support right there. It doesn't support what you're making your claim about. And so that can be problematic when you're working with it that way. So moving on. Oh, nice and highlighted here in this one. So what we're going to do here is we're going to use a workflow that we use in our lab. So before, this used to be a little bit more ad hoc. We kind of showed people a couple of tools and we let people, figure it out themselves. But instead, we've been actually working on trying to automate this process a little bit more so that it'd make it a little bit easier. And so that's what we're going to use here. So this is a link out to this is Elena's GitHub page where she has a phylogenomics workflow where we're going to do essentially what we're going to do for this next step, which is identify marker genes, perform alignments, trim the alignments, and then concatenate the files and construct a final tree. And so we're going to do all this using the examples that she has in her GitHub page. Okay, so this is where we get to the hands-on part. We're going to do it all in the next hour or so, maybe a little less, and then we'll be done. Okay, hold on. Did it to me last night. Okay, so if you are not there, we're going to move into CD Home, C-DEBI, Eco, Geo, Phylogenetics, and inside Phylogenetics, there's another folder called Phylogenomics Workflow. So we're going to move into that. If we hit LS, there's a bunch of information there. So our example genomes are going to be in that example folder. We have the hug marker list, which is the same, which is a list of just the names of the markers that we're going to be looking at. So it just looks like this. And then we have the same HMM database that we were using in the last session to look for these proteins. All right, and so what we're going to be doing is we're going to be identifying these 16 ribosomal proteins, so specifically large and small ribosomal proteins using the HMM models for each of these ones. So most of these HMM models come from PFAM, but a couple of them come from TigerFam. One resource that you can do and use if you'd like to find other organisms and specifically target certain subsets of marker proteins is this website called RiboDB. Which is a website that you can go to, and you can actually go and pick out organisms and select which markers you want from those organisms and pull those down and work for those individually. Okay. So the first thing that we're going to need to do for our data is we're going to need to move into example. If we look at example, there's a couple databases here, or a couple of directories here. We're going to move into genomes. So in here, we have 5 unknown sequences. Right now, we have both the FASTA sequences for the nucleotides, and we have the FASTA sequences for the proteins. I have done the prodigal step for this, right? So at some point, we needed to change the contigs to proteins. This has been done for you. When I did it this last time for this, it actually took a while. So we're just going to skip that step for right now. But you'd have to start. So we're going to start at the protein level of these unknown genes. And we're going to use a script that's part of that GitHub page called identify HMM, like that. Oh, let me increase the view here so we can see it a little better. All right, and then we can keep typing this out. So we're going to use a marker database, and that marker database is 2 folders up. So we're going to put in 2 dots and then another 2 dots like this to say where to look for it. We can tab it out as hug ribosomal markers.hm. And then we use a setting here called out prefix. And so you'll see what this does, but what this essentially does is it sets the name that we're going to use in terms of keeping track of our data as it moves downstream. So the out prefix here is going to be unknown. We're going to use as capitals. And then it asks, the last thing that we need to do is how are all of these protein sequences connected? And so we use the suffix here, .ffa, to say these are the files that you should be looking at to make these HMM searches for. So it runs a little bit. And then if we hit ls, what you see is in our output that we've generated is we now have a bunch of files where through those, from these unknown organisms, we have identified the different ribosomal proteins. And so what this means is if we look inside, so we do like less unknown, RPL14, we take a look in there, there are 5 sequences from these 5 organisms that all represent that RPL4 protein. RPL14 protein. And so that's the same for all of these. So we've pulled them out of these individual genomes. Okay, and that's pretty cool. But right now there's only 5 of them, and you can't make a tree with 5 proteins. So what we can do is we can combine them. There's an example set of proteins to work with in one of the other folders. And what we're going to do, the way we're going to do this is we're actually going to automate this in a loop on the command line. And so this is a new thing that we're going to use right here. So if it looks strange at first, don't worry about it. It's one of those things that we can do a copy paste. But first, let's type it out so we can all see what's happening here. So what we're going to do is we're essentially going to write a little bit of code here where Unix is going to run in a way that'll give us a bunch of results and read in something and then output based off of what it's read in. And so what we're going to say is while read P, we're going to define what P is. We're going to ask it to do something. So we're going to put a semicolon here. And we're literally going to say do. And the thing that we're going to say do is we're going to say do cat. So this is the command that we worked with yesterday. It's concatenate. We're going to take a bunch of files. In this case, we're gonna have a bunch of files that have some common name structure to them, and then stick them together and generate one output with a new name on it. So that way we can take the 5 proteins that we found for any of one of these particular markers and match them and pair them with the exact same markers, but for a set of organisms that are larger so that we can build a tree around it. And in this case, so we're going to do //hugref. That's the directory up and over one from where we're currently working. We're going to type in example. And so example set, there's a bunch of them in there. We're actually going to delete this RP part. And after this period, we're going to do open quotes, $P, close quotes, .ffa. And so this P, right? So while P, this is the variable that we're using that's going to change, but it has all the names here all have the same structure of example. P, and P in this situation is going to be RPL 14, RPL 16, all of the system, all of the ribosomal proteins that we're working with. So while it's doing that, take all of those sequences. When you find one, right? So in this situation, when you find reference set RPL 14, and if there's a matching unknown RPL 14, concatenate them together. So we're going to type out space unknown underscore, and then we're going to do open quotes, $P, close quotes, .ffa. And then we're going to output it into a new file, which we're going to call dataset1. And because we're still working with individual proteins here, we're going to do a open quotes, $P, close quotes, .faa. And then when you're done doing all of that, you're done. And we'll tell it explicitly that it's going to be done. And so to determine what variables P is going to be, we're actually going to cycle through a few of them. And we're going to do that based off of that list that's in the uppermost directory of this subset, the phylogenomics part, what I showed you really quickly, which was the hug marker list, which is just a list of those names, RPL 14, RPL 22, RPL 16, and it will read in each one individually, make this search, look for files that have those names in common, and then make a new file by concatenating them all together. So we're going to go up 2 folders to look for this, and then we're going to write hug marker. I'm going to hit enter, and then I'm ready for the barrage of questions that it looks like everybody has. That should run really fast. Does anybody have any questions about that? Yeah.
Yeah, what is the caret after done mean?
Yeah, so the caret, so if the caret pointing in the right direction, right, the one that we've been using for long, takes the information that we want and writes it into a new file, the caret in the opposite direction actually takes the information in that file and moves it into the process that we're working with. Great question. All right, so we can move on. Next slide. So now also, if we look really quickly, look at this. So now we have our unknowns that we've just generated, right? The first time around where we were looking for the sequences in our unknown genomes. And now we've combined them with some data set of genomes that have been set out as our reference genomes. And if we look at that now, so if we do data set 1 RPL 14, and we look at that, now there's a whole bunch of information in there. So these are the references that Elena has laid out to compare these unknowns to. And so right now we can see, if we look at the names of them here, that they're predicted to be types of alpha proteobacteria. Okay, so let's move on. All right, so now that we've done this in the directory that we're working with, we want to take all of this data set information that we have, and we want to move them up 2 directory levels so that we can work on it in the main workspace that we've set aside here. So we're going to use the move command, data set. We're going to put in the star flag. We're going to do period, 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 like that. And we'll hit enter. And so now if we hit ls, they're no longer here in our directory that we're working with. But if we go up 2 directories ourselves, they're now hiding out here. So this is just to separate them from their sources, right? So in the directory further down, we have a source for the references, and we have a source for the unknown proteins that we're working with. Now we can work in a space where, now that we have the data set that we want to work with, we can work somewhere else just to keep them separate, so that we're not going, where exactly is this information that we're looking for? And we can look at it that way. All right, so there are a bunch of options to do alignments with. There's things like clustral W, muscle, MAFT. And so which one you use depends on what you're trying to do and your computer and the size of the data set you're working with. So math is an example of one where they have a really rough alignment methodology that you can use that actually functions really quickly with a very large number of sequences. Muscle performs a really good local alignment using the whole sequence, while ClustalW performs a global alignment, which can lead to, in some cases, a number of gaps that may or may not be useful in downstream analysis. So in this particular instance, we're going to use muscle because we have a small number of sequences and we want a high accuracy alignment. So muscle performs an alignment and generates an aligned FASTA file as an output. So we're going to use the same example or same type of system as we just used, this bash while loop that we just did for the concatenating these sequences together. But now instead of concatenating them, we're actually going to run a program using them. And so we're going to use muscle, and we're going to use muscle iteratively. So each time we do an alignment, we're going to do the first alignment. All the RPL 14s will be aligned, and then we'll move down and do all the RPL 15s and the 16s, and you'll see that run. Muscle will run in each one individually when we type this out. So we can either type it all out or we can do a copy and paste. And not that we're pressed for time, but let's do a copy and paste. And we hit enter. This is muscle running. So that was 1. 2 is finishing up. 3 is going now. It's going on to 4. And it's going to go 5. It's going to go 6. It's going to go 7. I'm going to keep counting up because we're getting tired, and that's basically all my brain can do right now. And so this is going to run. It might be running faster or slower on your computer, but it will complete eventually. So that was mine. And I'm going to hit LS, and you can follow along when you get there. So now we have a bunch of files. So we have our dataset files we started with, which are the FASTA files, the FFA files, and now we've generated some type of alignment file to correspond with those. And so we can take a look at one. I think I want to look at data set RPL 16 for this example and their alignments. So you can see here, instead of a standard FASTA file, you have a FASTA file with gaps to indicate where the alignment sequences may or may not be. And so what we're going to do here is we're actually going to visualize these alignments in the virtual machine, right? And so we can get an idea of what it looks like when we have an alignment and when it has gaps in information, what we want to do in terms of removing areas that don't have meaningful phylogenetic information. And so on Wikipedia, there's a really nice list of 20 editors for doing alignment editing. Because the virtual machine has some limitations and we needed something that was free and we need to do all sorts of other things, we're actually going to use a program called JALView. And so you can call JALView by just typing in its name, hitting enter. And so it's going to generate this window. It's going to load up and it's going to open up a whole bunch of files and windows that don't actually, there are examples of it. Oh, look, there's a new version of JALView. Oh, look, here's an example of an alignment. And then there's going to be some more pause. Oh, here's an example of a protein. And here's an example of more stuff. And it's going to keep loading. And eventually it'll stop. And then as soon as it stops, we're going to break its little heart by Xing out all of this stuff. So these little X's. We can start with a blank screen. So this might take some time. JALView is very excited to show you everything it can do, and it might tax your computer a little bit. And so once that gets to the point where you have our blank screens, or we have a blank screen, yeah, cool. We can input our alignments from the file that we're working with. And so it says here, these are the ones that have been recently opened. But if we wanted to see all the rest of them, we're currently in file. So we want to look at anything that has this .aln format, because that's what we named our files to look for alignment. So here's all the options that we can look at. And so we're going to open up 2 of them to get an idea of the range of what we can work with. So if we open up RPL 14 first, we can make this a little bigger for everybody. You can see this. So here is an alignment that is actually fairly conserved across this group of organisms that we're looking at. So if we scroll down, we can see there's a lot of letters that all match each other. These bars at the bottom are giving us conservation scores, right? So these are just bar plots. So if the bar plot is completely full, it means that in every position of this protein is exactly identical. And so you can find areas that have slightly lower occurrence levels of always the same protein. And then we get a consensus sequence generated on the bottom. And if we go through, we can find areas that actually, we can find an area that has a gap. And so if we look here in the consensus, there's a place here where most of the sequences do not have an amino acid. And if we scroll down, you'll see that it's almost every position is empty except for back at the top, this Q. So there's this organism, this protein has a Q insertion here. And this position, because there's no other comparative positions to anything else for any of these other protein sequences, doesn't actually have a phylogenetic signature. So we can remove this one without changing the results of the phylogenetic tree. So this is really nice. This is an example of things on a very easy side of things. Let's try this. So we can add some colors to make it easier to see or less easier to see, depending on your preference here. So the colors of a block all indicate ones that are a really high similarity to each other. And so that's RPL 14. So there's only one spot that we can really see and identify. And if we were manually going to trim this, we would manually trim it there. We are then going to go up here, not inside that, and got another input alignment from file. And in this case, we're just going to take one of these other ones, which is RPL 16. And we can work with that one for a second. And here's one that has quite a few more gaps. All right, so here at the beginning of the sequence, there's this, it might even be the same organism, has 2 amino acids added here, which is causing a gap to form. So we could delete that to simplify the data. And then when we scroll all the way to the right, what we end up with is this area that has a lot of mismatch. Let me add the color here. Has a lot of mismatched areas. We don't actually have a really good alignment of this point. And so if we were working with this data and we were manually deleting or manually curating it, which we can be doing, but we're not going to, that we would say that this information here doesn't actually have a lot of phylogenetic meaning to us, even though there is one or two, maybe one positions that do have phylogenetic information. What we can do is we can either be really cavalier about it and just delete this whole area. And so we could do something like that, like this, where we highlight this whole region and delete it. Or we can get very specific and just highlight the areas that are mostly gaps. So we can delete that one, delete those 2, delete these 3. And now we've taken this area that had very limited amounts of phylogenetic information and reduced it down to just the positions that actually do have phylogenetic information. And so I keep saying, if we were going to do this, this is how we would do it, because I'm not a monster and I'm not going to make you do this 16 times. I could, you would all do it, but I'm not going to make you do that. And so what we're going to do is, now that I've shown you what the alignments look like and how you can work with them and double check them in a visual capacity, what we're going to do is we're going to use a tool that automatically or automatedly goes through and looks at alignments and makes a judgment call based off of how many positions have information and how many don't, and do the trimming for us. So this is a program that I think works really well for this. It's a very dumb program, right? It's not doing anything really fancy. It's just making a call based off of the alignments that we're working with. And so it's called trim-al or trimmel. And so this allows us to go through and actually automate this process. So instead of having to look at it. And so, you know, you're thinking, okay, I could have done that. So there's 16 ribosomal proteins. We would have had to gone through. There's only, looks like there's only a couple hundred. You know, I could quickly visualize all that. I could do that. So this gets very taxing when you get to very large numbers, a couple thousand genomes, things that are very distantly related to each other. All of a sudden, you're making lots of judgment calls about what's a good region to keep and what's not a good region to keep. And so this trimmel simplifies that a little bit. And once you have an output from trimmel, you can actually go back and take a look at it, and you should, and we will go back and take a look and see what trimmel has done to make sure that it actually makes sense biologically. But while we're at it, what we're going to do is we're going to run trimmel using its automated detection system, which is indicated there by the automated flag, in the same way that we've been running everything else, using this bash while loop. So it's going to again read in all of the names, fill in that P information, and run through the alignments and generate a trimmed alignment for us. So if we hit enter, this is going to run real fast. Cool. And so now we can go back. So first we'll hit LS, and you can see there's a whole bunch of new files that have been added. And now we can go back to JALView, which is going to open up again, and we're going to have a fun JALView opening party while we wait for it to open. This is what happens when we want something that's free, guys. It's gonna do things for you, even if you don't want them to do it. Yep. 2 questions. We'll go here.
Does JALView do the same as Meta?
Yeah, Mega will be is one on the list as an alternative for this type of information. Yeah. I have not. So the question is, have I explored any of these simultaneous? Is it trimming and alignment and trees? I haven't actually played with any of those tools. No. Have you? Okay. So we're going to really quickly just see how well trimmel did from our file. And so here we're going to have to set, looking at ALN. And so we're just going to go to that 16. So just so you can see, we're going back to that 16 RPL 16 alignment. And this time we're going to look at the trimmed alignment version of it. Let's look at that. And so really what we're really interested in is how did that work towards the end. I had a whole bunch of gobbledygook, and you see that it actually did a fairly good job here of removing some of those positions that had a lot of gaps in it. Great. So it's doing what we're doing, right? So it's what we would have done if we had spent a bunch of time doing it, but the robot did it for us. So that's really useful. And like I said, trimmel is really great, but it can make errors. And the more complicated the data set that you give it, the more likely it is it's going to make an error. And so going back and manually checking and making sure the alignments look meaningful and it's not throwing out lots of data and lots of information that would be useful is a good way of making sure to keep your sanity, to make sure you're not making a grave error in terms of the data that you're generating a tree with. Okay, so now we have we have all that information. So we have all of these separate alignments right now. All these separate alignments have now been trimmed. We now are looking at high quality, high phylogenetic information. And what we want to do is make the final sequence that we're going to use to make the tree. And we're going to do that by actually sticking all of these RPL sequences to each other in a version of concatenation, right? So instead of concatenating where we stack them on top of each other, we're just going to make a one long sequence. And that one long sequence is what we're going to make our phylogenetic inference off of. And so this example here is, so in these different files, these RPL files, RPS files, you have sequences with names. And so in this situation, sequence 1, it'd probably be sequence 1, slightly worded differently than this, but you have RPL 14, you have sequence 1 in it. It's all one sequence. You have RPL 16, you have sequence 1, it's a completely different sequence. And so what we want on the output is taking these two sequences and sticking them together. And we're going to do that for all 16 of these ribosomal proteins. And so we're going to do that using a script that's part of the Binsanity program. It's called Concat. That's what it does. And so if we type out what we see here, we're going to do concat-f. So it's asking where are we going to work? We're going to give it a period because we're working in the directory that we're working in. -e, what are the extensions that we're working with? It's going to be called .trim.aol. So it's going to look for all the files that have the same extension name. We're going to give an output name, prefix, and that's going to be still dataset 1. And then we're going to call, the prefix is the input information. So now it's going to be looking only for things that have trimmed ALN at the end and dataset at the beginning. And then we're going to print the output. And so the output for this, we're going to call dataset 1.hug ribosomal.trimmed.concat, right? So we're adding a lot of information. So if we come back to this, we know what's been done. And then we're going to add a little information here. And what this is going to say is, so there are 16 markers that we're looking at. Only include organisms that have at least 8 of those markers, right? And so the reason behind this is that maximum likelihood is actually can work fairly well with missing information. So if you have an organism, and especially when you're working with metagenomic bins, you might have organisms that are partially incomplete. You might not have all 16 of these markers. And so we don't want to throw out that information because we can still figure out phylogenetically where they belong. But at the same time, the less and less markers you have, the harder and harder it is to accurately place that genome in the data set. And so we pick 8 because it's the halfway point. So it has half of the data, but is missing half of the data. You can play around with this a bit. You can make it a lot more stringent. You can make it more relaxed. And it will give you slightly different results depending on the data set you're working with. If you're working with closed genomes, and you want to make sure that you're working with genomes that are, for whatever reason, maybe you're working with closed genomes and you got them from a source and you can't trust all of the outputs, you might want to have a really high number here. Maybe you want 15 of the 16 or 16 of the 16 to be present because you're supposed to have them all because they're closed. If you're working with really incomplete metagenomic bins, right? So something that's in the order of 30% or 40% complete, you might be a little bit more forgiving about which markers it has and doesn't have, and you can make it a little lower, maybe 8, maybe 6 or 4, something like that. And it will impact the tree, but it will still give you some information about where those organisms are on the tree that you're working with. So if we hit enter here, it'll run really quickly because all it's doing is looking at the names and sticking together. Yep.
How does max likelihood correct for missing information?
How does it do it? So what it does is it just doesn't calculate that portion of it for that one particular sequence. Okay, so I mentioned these really briefly. So there are lots of ways to build these maximum likelihood trees. RaxML is one way of doing it. PhyML, we're going to be using FastTree because it's fast. And the difference between it is that a RaxML maximum likelihood tree actually adheres to very strictly the models and the mathematics behind maximum likelihood in terms of calculation. FastTree is very open about this, calls itself an approximate maximum likelihood tree. So it makes a maximum likelihood calculation, but it makes a couple of assumptions along the way that speeds up the process as it's running. You can implement similar types of models of evolution and things like that. The thing about FastTree is, and it depends on who you talk to. The FastTree guys and gals think that they have a system that works really well. It approximates a maximum likelihood tree pretty well in the high 90% variable range of it almost is identical in almost all instances. People who spend a lot of time thinking about phylogenetics would say, maybe not. If you really want to make a case, and I talk about this with the undergrads in our department all the time, is that if you are making a case, a strong phylogenetic case where it's really important where the branch point is and where they are in relationship to each other, then you need to do something that's a little bit more robust than FastTree. So RaxML is the way to go to that. So if you're making a phylogenetic argument, you have to use a phylogenetic tool. If you're using it to, get some information, the ideas of like, in this case, what we're looking for is some general relationship, so we can assign some name to some unknown organism, or we're looking at, your first attempt to find out if your alignments or your sequences are related to each other. FastTree is awesome because it's fast. It's just the way it is. So we're going to do that. We're going to construct a maximum likelihood tree with FastTree. And so that's built in here. So we're going to do FastTree like that. We're going to use 2 different flags to set the types of models we're working with. So we're going to do gamma, which is this correction. It's a time-based correction. And then the model we're going to use is LG, which is not listed here for some reason, but I can give you more information on it. I don't think it's on the next slide, but as we go along. So we're going to do dataset1.hug, and that should open up this whole thing. And then we're going to output the results.hug into, we're going to call it a NUIC file. So this is the type of file. It's a file that has all the tree information in it. And so when we can look at this, and what it is really just information for a visualization tool to look at, and it tells you all the distances between all the points. And so when you look at it, it just looks like a bunch of numbers with parentheses. Yes.
What does the gamma do?
Yeah, so the gamma is a time corrected flag. So it allows it to, after it's made all of its calculations, it makes a final adjustment to make sure that the calculations are correct. And we can actually bring that up. If you do FastTree by itself, if you've not typed it all out, if you type, it actually will explain a little bit what the gamma flag and the LG flag do. We can do that together. So if we hit enter, it's going to run. No, it's not. Error reading header line. What have we done? Did everybody get that?
Yep. Oh, nope.
Oh, not that one. We don't want that. Oh, it's because it's empty. Do I? Great. Let's see. All right, let's go back up. We're doing it live, folks. So this ran, right? The output already exists. We do not want to overwrite it. All right, so that is definitely empty. So we're going to remove that. And we're going to,
I see what it is. So in the concat, you had a capital S instead of a lowercase s for dataset 1.
here we go. Did everybody copy that along with me? So who worked for it?
It is working.
It is working. So some people it's working for. Great. Good job, everyone. Don't make my mistakes. So I'm going to just copy this and paste it. There we go. And then when we run that, there you go. Now it's running. So you should get something that looks like this. It's going to take some time to run. And then before we look at the results, we'll run FastTree without any commands, and we'll take a look at some of the other options that are part of it. Okay, so output like that. So if we run FastTree by itself, we can look at some of the parameters that are here. Give some information. So if we scroll up, so there's a lot of different ways we can input this information. So FastTree works not only with proteins, but it can also work with nucleotides. You have to be explicit when you're working with nucleotides though, because it's assuming that it's a protein file. There's a lot of information that we can keep. We can keep the log file. We can make it run faster than it does already, despite it being a fast tree. I was saying that, so we expressly wrote out that it was the LG model. So this is a amino acid model, substitution model. This is a paper that it's referencing, Lay Gasol 2008. There's some other options here, like the DASH-WAG, which is the Whelan and Goldman, which is a different model from 2001. If you're working with nucleotides, you can do this generalized time reversal model. So that's nucleotides only. And then so gamma is down here and explains it here. After optimizing the tree under using a specific rate category site approximation, it rescales all the lengths to optimize the likelihoods of the tree. Okay, and so we'll take a quick peek at the output here from hug ribosomal concat NuWic. Right, so this is what a NuWic file looks like, and it's going to be the tree information here. It's going to look like this. It's a little scary. And so what this is showing us, like I said really briefly, is so if you were to actually go through here and look at this all a little bit manually, and it's a little hard because the names are fairly long here. Every set of parentheses is giving some distance information between 2 points, right? And so if you find 2 parentheses that only have 2 items in it, and then a number, it tells you how far apart those 2 names should be on the tree. And then when you import it to a tree program, it is able to draw a line that solves that problem for you. There's also information here about your bootstrap value, which will be displayed on the tree as well, so that all that information is there. So, you know, this is what it looks like. You don't have to know what it means, but it's good to kind of have an idea so that if you run something and it doesn't work, that it might be, oh, okay, so let me double check to make sure my NuWic file looks correct. Okay, and so then we've run fast tree. And now what we want to do is actually visualize the results here. And so we're going to use a program that's in the virtual machine right now called Dendroscope. That should open up like this. We can get rid of this little error or message bar. So I'm going to make this big. Like that. And if you hit this little folder button up here in the corner, we can look for a NuWic file in the directory that we're working in, like the one that we just made, and open it up. We're going to tell it to interpret our labels as bootstrap values. And there we go. So these are going to be our results. So right now, the default output is this cladogram, but really what we want to do is we're going to look at it with some actual information in terms of phylogenetic distance. So there are these different buttons up here that you can push. The first one on the left will do that. It resizes everything. So now that the distances here actually mean something. So in this version here, we just had to fix everything. Every time there's a split, it's a set distance, and we can kind of see some relationships here. In this version of it, we're actually seeing every distance length here is actually meaning in terms of how far apart these sequences are from each other. These numbers that you're seeing at each of the points or at each of the branch points are your bootstrap values. So this is out of 1,000 or a fraction of 1,000. So what you're seeing is in this example, so right here, this point in 997 of the trees that were run of these 1,000, this point always existed in terms of the branch of these organisms here and the branch of these organisms here. Conversely, the branch point here is much weaker. Only 247 of the trees had this branch point. So in a bunch of these other trees, there was some other slightly different relationship between these 2 branches, the phylogenetic tree. And so we can do things like we can expand it so we can see more details. And so you can see there's a lot of genomes that we're working with here, all of them within the alpha proteobacteria. And if we scroll down and we look and see where our unknowns are, so unknown 1 and 3 are most closely related to this Acetobacter. What is this? Pasteurinaceus and Pasteurinaceus. And then this one here is a Rhodospiraliaceae, Acetobacter aceti, right? And so our genomes, in this particular case, it looks like the genome unknown 3 and the genome from the database, that's basically a flat line right there. So that's indication, yeah, the indication that the sequences that we're looking at are 100% identical. And so this might be an indication that these, I assume that this Acrobacter Pastor Ionis, saying that definitely wrong, is probably a cultured isolate, while the unknown 3 is an organism that came out of the Tara Oceans data set, right? So the chances are they're not actually exactly the same organism, but they're probably strains of each other. But in this situation, you probably could get all the way down in terms of accurately calling the species on that organism. Unknown 2, there's a little bit of a branch here. So this might be, again, another strain within the species Acrobacter aceti. So that's unknowns 1, 2. And then down here, in the much more problematic area, we have unknowns 4 and 5, which are closely related to each other, but don't seem to have any relationship to any other known alpha proteobacteria, even though this group is fairly distantly related to it still at the same time. So we know they're within the alpha proteobacteria, and we know a little bit better if we did something like add an out group to anchor the distance of the tree, compare this to other organisms that we know are more distant related than the organisms that we're looking at. But what we have here is some alpha proteobacteria that, at least based on the references that we're looking at, doesn't have a known entity in the reference databases. We'd have to ask Elena to double check. So are they really unknown and don't have?
No, those are known.
They're known. You just didn't include their references. Yeah. Okay. Great. Cool. So that would be how you look at the tree. One alternative to, well, not one alternative. There are lots of alternatives to dendroscope. Oh, I want to actually quit out dendroscope. Yeah. One of them that we actually really like working with is the interactive tree of life, which is this link right here, which allows you to import your NUIC file into your browser window, and then you actually get the tree presented in your browser. And they have lots of ways of adding information to make your tree have like some more meaning to it, right? So if you wanted to add little elements to show, if you're looking at genomes and you had elements that you wanted to include there, you could add those. If you wanted to create color ranges to say, these are all the acetobacteria right here, and we can color that range. It's a really nice tool to work with, much nicer than dendroscope on the grand scheme of things, but dendroscope works when you want to work on something that's local, where you're not connected to the internet. And so we're wrapping up. This is the last couple slides. We're going to wrap up about an hour early today. So those of us who are catching a flight will get to leave without missing anything. That's pretty exciting. Everybody else will get to start drinking a little early. That's fine too. Happy Friday to us all. And so in summary and closing, what we did was we identified marker genes and chose based on the distance of the relationship. We performed alignments. We trimmed those alignments and we visually checked them. We concatenated the alignments. We didn't have to check the names because all the names were correct. And we constructed a phylogenetic tree and then looked at it, analyzed it to make sure that it actually made biological sense, right? We didn't see any crazy information there that, they weren't, the base of the tree, we weren't missing some number of sequences on it, which is really important to do. You never want to do this all in a vacuum. So some of this is listed in the references and citations, but also here in the presentation. So these are direct links to different programs that were mentioned as part of this. So ARB and Silva are both, one is a tool, one is a database. We've probably heard of the Silva database for 16S rRNA sequences. If you want to start playing around with Bayesian trees, Mr. Bayes is one of the more commonly used and historically accurate Bayesian tree programs. There are other alignment tools you can work with, including Genius, which is not free, but is fairly useful. And then lastly, but not least, ITAL, which is good for your tree visualizations, interactive tree of life. So I'm going to hit X here. I'm going to close down this. I'm going to close down that.
You know if the hug tree was with that ITAL? Like
If they visualized it?
Yeah. Or like how they generated that particular.
I don't know off the top of my head. Thank you, YouTube, for following us. We're not